In Chapter 7, we're going to be translating mRNA into protein, and that's going to let me talk a little bit more about functional programming. And the idea here is that DNA makes mRNA, and we've already talked about that. That was one of the early chapters where we changed all the T's in a DNA string to U's. And now, given that mRNA string, we want to change that into proteins. And this is quite a bit more difficult than just swapping T's for U's. And if we look at the Rosalind PROT page, it has a little bit more on the background. It's fascinating stuff to read about, but we're going to get into just this. The problem here, we need to change this string into this string. And we're going to do that using this RNA codon table that they provided to us. So when we see a string of 3 U's, we're going to create the string F. And on through this entire table, a few that are very important are these stop codons. So these 3 codons will tell us to stop turning this into protein, and we're going to need to find those. And so many computational problems, pretty much all of what we do is just turning one thing into another thing. And here we're going to be changing one string into a different string using a substitution table. And since we've talked about tables before and kind of things like that, one of the things you might, hopefully, your brain goes right to is a dictionary lookup, a lookup table to translate one string into another. So we're going to be using lists, for loops, list comprehensions, definitely dictionaries, and I want to talk some more about higher order functions. I really appreciate these, and I feel like once you've gotten them into your brain, it can help you find a lot of shortcuts in programming. In the end, though, I'm going to go back to Biopython. There is a built-in function to do this, and so it's actually trivial. But there's still a lot of value to be had in figuring out how to do it on your own, especially because we're going to be talking about how to extract the codons manually and how those are related to something called k-mers, which is something that is used quite a bit in bioinformatics. And that is going to allow us to talk about using slices, which we've just talked a little bit about before, and then we're going to lean more into that on this one, on this problem. You can take slices of strings or lists, and if you'll recall, strings and lists are kind of interchangeable in Python. We're definitely going to talk about a dictionary as a lookup table. We've seen several times that a for loop can be turned into a list comprehension, and that can be turned into a map expression. So we're just going to look at the relationships between those. I have also introduced filter in the past, and I'm going to talk about how a similar function called takewhile is like filter. And sometimes those lambda expressions can get quite long to type, and I don't really like typing out the word lambda. So I'm going to show you here how to do partial function applications by using the function called partial from itertools. And finally, we're just going to go back to our good old trusty Bio.Seq module that will do all the work for us. To get started, go into the top level directory of your Git checkout and make sure you do source v&v bin activate, and we are using UV, again, a UV virtual environment for this exercise, just as in before. And as with so many of the previous exercises, I'm going to have 2 windows open, so I'm going to do that in both windows. And I'm going to go into my 07 prot directory. And the first thing I want to do is just copy solution 1 to a program called prot.py and then run this to see what are the inputs and outputs. So what we see here is we want to translate RNA to proteins, and it takes as a positional argument an RNA sequence. So if we were to go here to our Rosalind example, I always like to use these. Instead of passing -h, I'm going to pass that sequence, and I see that I get mama protein string out of this, which is what is the expected output for this given input. And this is how this works with Rosalind. If you haven't actually been solving the problems here, uploading your solutions, it's a lot of fun. You get to go through these and see them passing. And so they give you this sample data set, and then you figure out how to fix this one. And then when you go to solve it, they'll give you a much larger data set that if you have not actually written a program to do it, you probably will not be able to answer in the 5 minutes that you have to upload an answer. So given this input string, we get that output string. Pretty straightforward. That's what we expect. And if I run make test on this at this moment, we see everything is passing, and that is what I expect. So if you think that you can solve this, I again would challenge you to first try this on your own. There is so much to be learned from figuring it out on your own. Even just coding along with me teaches you a lot, and I do encourage you to be typing everything that you see me typing. But even without that, I think that there's a lot to be there's more to be learned by doing this on your own. So to get started on this using new py, I'm going to use this command here to create my own version of prot.py, and it is going to have my all my arguments as normal. I will not be using text.io. I'm going to create a single input string of RNA, and I know that this is really overkill to use all of this infrastructure for such a simple program, but I think it builds a lot of muscle memory to do this, and short programs tend to become long programs, so I would always start from this highly structured program to even make a simple program. So here this would be, I think I'll call it, what is it, input, just RNA sequence. So RNA sequence, and that is all we need, so I can get rid of all the other arguments. I will parse the args, and then I will return RNA is equal to args.rna. And with that, I think it's always good to just start off by just printing what we have, print args.rna. And with that, we'll go over here and we'll just run this. I can go back and run it with, whoops. There we go. Oh, no, So the problem is that I was running my program. I had actually created it in the root level of this, and so when I was running this over here, I was still running the example program. What I need to do is move the one that was up there into this directory, and now when I run it, I am just seeing the protein string, which is what I expected to see. Whoops, I need to be in the 07 directory, and then now. So this is the program I'm working on, and this is a good place to start. And at this point, if I do PyTest, always good to see what's happening so far. I'm already passing 2 of my tests because I've created the right program and it has a usage. So that's great. So moving forward, what do I need to do? Well, the first thing I'd like to talk about is k-mers and how they relate to codons. So we have, I need to go through this string of RNA and read it in groups of 3s to create a codon. So as I'm going through, I need to grab 3 bases and then move on to the next 3, and they do not overlap. And I have a little picture here of what that looks like. So given this string of AUG, that's the first codon, and then after that GCC, and then after that AUG. And if I, as usual, I'm going to go into my REPL, and let's just put in this RNA string here and talk about some techniques for doing that. So given that RNA string, we've talked a little bit about slicing up to this point, and we used a particular way of slicing in order to reverse a string in an earlier chapter when we were doing the reverse complement, where we did -1. We did not put in a start or a stop position, and we put in a step of -1, and that reverses the string. So we've already seen that we can do something like if we leave off the stop and say I want up to 3, then it's going to give me starting, if I leave off the start position, it's going to start at the beginning of the string, which is position 0, and it's going to go up to but not including 3. So which is actually the 4th character, but it's the 3rd offset. So that is the way to get the first 3 letters of the string. And so it's probably better here for me to do 0, 3, and then if I go 3, the next codon is 3, 6. And so I was a little bit confused here, because when I looked up, I was looking at this string, I was thinking, hey, that's not the first 3 characters, but I had to remind myself that is the reversed string. So I'm going to print RNA here again so that I can see it. And there's the first 3, and here's the next 3. So we need to see, is there a pattern emerging here? And what the pattern is that we need to step by 3s for the starting position. So 0, 3, 6, 9, and so forth. And that will be the start position. And the stop position is going to be whatever is the start position plus 3. And so how can we get all the start positions in a given string? Well, we've seen range, the range operator. And if I do range 10, I'm going to get range 10, which is totally not useful. So I need to force that in the REPL, and I need to coerce it into a list, which I can do with the list function. So given that, that's useful. There's also something else to know, is that range will alternately take 2 arguments, the first of which is where to start, and the second of which is where to stop. With only 1 argument, it's assumed that it's going to start at 0 and it's going to stop at that top number. So that's a way to get a limited range. So I can actually give it a 3rd argument, just like with the list slicing, and that is the step size. So for instance, I could say range from 0 to 10, stepping by 3. And so now I get 3, 6, 9. Very cool. So how can I use this with my RNA string? Instead of hard coding 10, I will type go to the length of RNA. And those are all the start positions for the codons in the first reading frame. So if you know your biology, you know that we don't always have to start from 0. Sometimes we can start from 1 or from 2. So that is how you get your different frame shifts. So this is, we're only going to talk about the first frame, and so it's going to start with 0, and that is the way that we get the starts. So we can use a list comprehension now, turn this into, let's just take this step by step. Instead of saying list, I'm going to do a list comprehension of in for in range, and saying that out loud using the variable in for number here makes it confusing to say it out loud with the in word, but since that's the variable name I chose in the book, I'm going to stick with it. So this is another way to write the exact same list operation, doing a range. So just doing list range gives me this, but this is the way to write it as a list comprehension. Well, I can take that same value, that same expression, and instead of returning a single value, I can return a tuple of n and then n plus 3. And it's quite a long list, so let me just do the first 5. And so I can actually do a slice of that list expression, that list comprehension. And so it shows me here that I need to go 4 RNA starting at 0 and going up to 3, and then 3 to 6, 6 to 9, 9 to 12. So using that instead of returning a tuple, let's take that idea and say what I actually want is RNA starting at n and going to n plus 3. So I'm changing the tuple into a list slice, and since that's also going to return a long string, I'm just going to return the first 5. Those are the first 5 codons in our RNA string. It's also our first 3 mers. So when we talk about k-mers, the k is some size here, and mer means a share, like in the word polymer. So we often refer to k-mers by their size, so these would be 3 mers. But the difference here is that k-mers actually overlap. They overlap. So in the first 9 bases of this RNA sequence, there's actually 1, 2, 3, 4, 5, 6, 7 3 mers. And we can actually find this using this formula. The number of k-mers in any sequence is the length of the sequence minus the size of k plus 1. So if we look at what is the length of our RNA sequence, we can find, I need to establish k is equal to 3, sorry about that. And if I say what are the number, there are actually 49 k-mers in our RNA sequence that we're dealing with. And we could get all of them, let's see, but we are not trying to get the k-mers in this instance. We only want the codons, and so we will be talking about how to get k-mers later. But it's good to know that basically codons are non-overlapping k-mers, and so the number of codons that we have in our entire list here, we can do a length on this list comprehension, or we could assign it to a variable, obviously, and then take a length of that. So codons are non-overlapping 3 mers. Now, how do we turn those codons into proteins? And so we've gotten this handy dictionary structure, and hopefully at this point you can see how to do this. I would encourage you to stop, code it yourself, and see if you can pass the test. But if you want to stick with me here, I will keep walking you through this. So a dictionary is a really good way to code this up. It's certainly not the only one, but it's probably really the most convenient way to do this. And so what does that look like? So in I need to come up with some sort of codon to, I created a dictionary in my code called codon to AA for amino acid. And so what I'm showing here is that given this dictionary that has this information in it, I can take each one of these codons and go look it up in my table, and then I can append it to my list of amino acids. And I'm showing here that given this this dictionary, that this is how this string would be converted into proteins. And you can see what here I have as a list, and hopefully at this point you look at that and immediately know how to take a list of characters and turn them into a string. You probably should also be thinking, well, you know, I could have actually just initialized this to a string, and instead of doing append, I could have done a plus equal and just added the next the next character onto the string, and then I would have ended up with just a string of these characters. And so however you want to represent that is just fine. So this will get you through. If you can figure out how to set up that codon to AA table, you can put that into your solution, and that should pass all of your tests. So I'm going to give you another chance here to pause the recording and go code that yourself. So I'm going to go ahead and continue. I can actually take this bit of code, even though it's a little ugly. I can go paste this into my REPL, and the next thing here is I can run through this code here. This is what is in solution 1. So let me look at solution 1 real quick, and there is all my code. I can set k is equal to 3. Let's see. I wonder if I can. What I can do here is a little trick that I've learned. If I push all that back to the left side, I can actually copy and paste it straight in here. And so let's see what protein says. I have now come up with my mama protein string. So what is going on here? We start off by saying k is equal to 3 and our protein is equal to the empty string. So this is the other part of the solution I was talking about. So for codon in this list comprehension that we wrote to get out all the codons using our range operator and then using this string slicing, I get each codon and then I use my codon to amino acid table to get that codon or a dash. So we have to account for the fact that we might get bad input and we don't want our program to blow up. Remember that this codon to AA table, it is just a dictionary. So if I ask for, XYZ, it is going to blow up. It's going to create a key error that is an exception, and our program is just going to fall over at that point. So if I were to ask instead for .get and then put a default value of say dash, then I would get, there we go. I will get a dash out for any unknown codons, and so that is much safer code. So I'm going through, and instead of doing the append operation that I showed with a list, I'm doing this plus equal to add this character to the string. Now, one thing before I add that character, I need to see if I've reached the stop codon. So in my table, I chose to represent the stop codons with a star, which is very common that you see in representations like this, because the word stop, it's 4 characters instead of 1. All the other ones are 1, so we really want to have a single character to represent a stop codon, and it's pretty typical to use star. It's also pretty typical not to include the star. The star just means stop transcribing, stop translating, sorry, and so we just would stop here. So you see here, if the amino acid is equal to the star, we don't actually append it. We would break. And I'm not sure if we've really talked about break, but it is a statement here in Python that says we are in this loop, and so break out of the enclosing loop. And so if you have multiple levels of loops, if you have a for loop inside of a for loop, the break will break out of the one that it's most closely related to. So if you say break on the inner loop, it'll only break out of the inner loop. So this is a way of stopping going through this. And if we had a very, very long protein string, like say the entire human genome, and wish you probably wouldn't, but if you had in a huge input string, this would only go as far as the first stop codon. And we're going to see that some of our other solutions don't immediately stop at that, and so we actually end up kind of doing some extra work. So I would say that this is a very straightforward and very efficient way of doing this translation. And with that, we should be able to pass. So this is what our solution looks like. I'm going to copy solution 1 to prot.py, and I'm going to do PyTest to ensure that this does indeed pass all the tests that we expect. You can do make test as well. It will run a couple of additional things like flake8, and I am in the process of commenting out the tests for pylint, which don't seem to work very well anymore in MyPy. Choosing instead most of the time these days of using TY to help me check my programs, and it can catch things that flake8 doesn't, and so I kind of triangulate on a few different tools. So in my second solution, I would like to, as with a few other programs in the past, there's a lot. I would like to move some of this code out of the main function into their own functions so that I can then have a test for them. And I can come up with a couple of functions now. The first of which is creating codons. And this seems like it's a useful thing to do. It seems like in bioinformatics we do this kind of thing a lot. And so it would be really good to put that into a function. So let me define my function here real quick. And the idea here is when you're writing code, I think it can really be helpful, first off, to use type hints, because then you're kind of saying out loud to yourself, okay, I'm going to take a thing, a sequence, and what is that thing? Well, it's a string. What do I expect to get back? Well, what I want is a list of strings. And it could be an empty list. If you're passed an empty string, you're just going to get an empty list. But when it is something good, the things in that list will be strings. And so in order to use these type hints, I need to go up here and import list. And so already I feel like this is kind of giving me an idea of how I want my function to work. And notice here I haven't done anything else with the program. I'm just adding this function, and I'm thinking about how do I want to use this. And I really find that if I write my test first, it really helps me to think about how I want to write the function. So I'm going to copy and paste this test here, and think about this. So when I pass the empty string, which if I'm expecting a string, I always start off by passing the empty string. What I expect to get back is an empty list. And again, it's up to you how you want to code this. Maybe you want your program to throw an exception when it's given an empty string, and that's your choice. However, you're going to then have to learn how to deal with exceptions properly. And so it's going to be a fairly simple program. It's going to return incomplete codons, not program. It's going to be a fairly simple function. So given a string like A, it's just going to return A. Given ABC, it's going to return ABC. Given ABCDE, it's going to return ABC and then DE. So some of these codons are actually going to be incomplete, and that's fine. In my translation step, we are taking care of when we try to get a codon that doesn't exist, it's going to return something else, and we'll be able to filter those out. So this function is going to be very limited. It's essentially just going to return this bit of code. So how do I now code this? Well, I take this idea here essentially from my main function, but I'm going to take the bit here that I've written. And now I've written my codon function, and now I want to see, does it work? And I can do that by running PyTest. I'm going to open a second window here because I'm going to keep that REPL open. I'm going to go into my BioFX. First thing I'm going to do is source my V&V, bin, activate, and then I can go into my 07, and then I can run PyTest, and I can run it just on prot.py. Because what I'm trying to do is just run this unit test. I'm not even using the function yet in my program. I just want to see, does my unit test pass? And it does. And just to be clear, let's go in and mess it up. Let's make the codon size of k is equal to 2. And now if I run this, I'm not getting the right output, right? It passes the first couple tests, but when I ask for ABC, I'm saying it should be equal to ABC, and what I'm actually getting back is AB and then C. And that is not the same. So I can see that my test is working, and my code is working, and that's great. So moving forward, the question is, how do I incorporate this? And I have one more note here. Since there's 3 lines of code here to build up the return, and then do this for loop and append the codon to the return value, and then to return the return value, it's actually much nicer to have it all in one line, return a list comprehension of the for loop. And so my code gets much, much tighter now. Visually, that's just easier to see. And does it still work? Let's find out. Run our test on it, and it still works just fine. So this is, to me, a nicer way of writing that, and it also gets around, first off, it's shorter, but it also gets around having to modify a value. I prefer, this would be a much more functional programming approach to solving this. So I appreciate this better. So how can I incorporate this into my program? So instead of, this is the code right here, and I can call it by saying codons on args.rna. And so now my main function has gotten a little bit easier to read. There's still quite a bit of stuff going on there, but if I run pytest xv, I should still pass all my tests. So I can see that I know from a function standpoint, from a unit standpoint, that this function works. And now I can see from an integration standpoint that when I put my function into my program works. And that is the idea. This is one of the ideas I'm trying to drive home with this book, is that we can decompose a large problem into small problems that we can solve and think about, and then we can compose them together into larger programs that work reliably. So one of the things that's kind of cool about this, and that I really want to stress, is when you've written a function like this, it now has become something that you can share among other modules. And I left my REPL open here, and I want to show you now. I'm going to import OS real quick, so I can say OS lister on the directory that I'm in, and you can see that I'm in this directory here. I can actually os.get cwd to see my current working directory. So I'm in this 07 directory, and if I do a list, you see in here my prot is right there, and so I can actually import functions. So from prot import codons, and I now have access to this codons function, and I can convince myself it works by passing it a string. And that's super cool. So the thing that's going on here is that there's not really much difference for Python between a program and a module of code that you can write. So essentially what we have here is just a Python module that in some cases we are actually executing. And that's what's going on here at the bottom. When we execute this block, this file of Python code, when we run it like a program, then the namespace, the main namespace, is equal to __ main _. And these are called double under variables. They're these special kind of hidden global variables in Python, and I always hate to talk about this part with beginners because namespaces and things like that don't really make sense, but that's what's going on. When I execute this, the namespace becomes double under main double under, and so therefore if this is true, then I say go execute the main function. And so what happens is that when the operating system sees the shebang line, it says, oh, I'm supposed to use Python 3 to interpret this file. And so then it executes Python 3. Python 3 then ignores this first, ignores that part of the file, and goes through and starts reading. And it reads down, reads the class definitions, it reads the define for get arguments, it reads the define for main, it reads the define for codons, and all of this. And then it gets to the very bottom and it says, it executes, it reads this, and it says, oh yes, in this instance, while I'm executing this, the main space is equal to double under main. So, oh, okay, now go back to the top and execute main. And so that is how our program starts. And when you import the program, when you import the module, this is not true. The namespace is equal to prot. And so it's a different namespace, and so we do not execute main. And so if you don't have this check at the end, when you say import prot, it will automatically go execute main, which is not what you want. So it's really just kind of a little hack inside of Python. That is how a module of code can become an executable, but it still doesn't change it from being something that is still a module of code. And Python has no idea of private variables or private functions, so any function in this file can be executed from outside of the file. I specifically imported just the codons function, but I don't have to. I could instead just import the entire module, and then I can call prot codons directly on using the module name and the function name. And so it looks like that. And so that is another way to run it, to import it. I can also import as, PR if I want. If there's some sort of a name conflict between, my module name and some other module name, I can import it as an alias and then use the alias like that. So maybe a little more than you wanted to know about Python modules, but the point here is that when you start writing functions that are generally useful, then you can put them into a Python module, a .py file, and then you can, from other programs, if they are living in the same directory, very easily import those functions and execute them. So that's actually really useful. I encourage you to think about writing these kinds of functions and then sharing them rather than copying and pasting them in your different programs. So how can we use some of these ideas to go forward? So we have a Codons function, and it has made our main function a little easier to read, but there's still a big block. This whole translation table is still part of my main function, really taking up visually a lot of space, and I feel like that belongs in a function all by itself too. And so I'm thinking about having some sort of a translate function. And so for this, I'm actually going to go over to, I think it's solution 3 here, is that right? And yeah, so there's going to be this translate function that I'm going to write. And the first thing I'm going to do is think about, I'm going to paste in here a test for it. So given some translate function of the empty string, I'm just going to have it return the empty string. When I give it AUG, I want it to return M. When I give it this string of DNA, I want to get back MP. And when I give it this longer string here, I expect to get back our mama protein string that we've been seeing. A little trick that's going on here, because this is such a long line of code, I could have written it like this, and then it gets into this long line that I don't really, it can be difficult to break long lines because of the way Python is very sensitive to indentation and spaces and line breaks. And so one of the things that you can do is in like a list context, which is the case here because it's in these parentheses, in certain contexts, adjacent literal strings will be concatenated into a single string. And that's a really interesting edge case. The first time I came across it, I thought there was something surely was going horribly wrong. But no, this is actually this is a feature, not a bug. And it has come up to bite me in many different ways. Like if I wanted these to be two different arguments, and I meant to have a comma here, and I forgot the comma, then I would actually be sending one argument, very long string, instead of two shorter strings. And one of the places that it's really gotten me, I will show you here, is if I had, say some sort of an option here, like for an out file. And let me put it on separate lines the way that I would probably do. And let's say it's -- out file and -o and -- out file. And I meant to do this, which is the way that this is usually written. But if I forget that comma, what happens is that Python will concatenate these two strings into a single string, -o -out file. And that is so very, very not helpful. And but that is the way that Python is intended to work. So given that, I can sometimes use that to my advantage. And if you don't know what is going on here, that would be extremely confusing to you. But it is the automatic concatenation of adjacent string literals. Okay, that was a long-winded explanation. But the point is I want to get into moving all of this code that from the main function into a translate function. And so if, to start off here, I could start off by writing a translate function, that is going to take some, I think I'm going to call it RNA, that's a string, and it's going to return a string. And I can just, for the moment, return the empty string. And I can make sure that my test is failing. Let me go use my other window. If I run PyTest on my protein function, I am indeed failing my test. So I need to now write this function. Well, it's going to take this code here. I want my table now, and I essentially want to move it into here. And the way actually I wrote this in this solution looks a little different than this. I'm going to use this version that I wrote. And first off, let me just make sure that this works. That works just fine. And so let's just talk about this solution. What's going on here? So I use my list comprehension, and now my codon to AA table is defined here inside the function, and so I use my list comprehension that we saw before. And if there is a stop codon in the AA list, then I'm going to truncate the AA list to the index of that of the stop codon, and then I'm going to return AA. So this was I mentioned earlier. This solution is going to go all the way to the end of the string, and it's going to read right past any stop codon, and in fact there could be several that it may find. And so in a way it's inefficient because it's doing a lot of translating that we don't need to do, and then it's going back and truncating the list to wherever the first index of the first stop codon is found. And that's okay. We'll see that the Bio.Seq.translate does the same thing, and so it's fine. These we're not really going to be translating such huge strings that it's going to present a problem. So let's just break this down. What's going on here? It's going to turn. So in the REPL here, I'm putting in my codon table, and I'm creating this codon to AA, using that to get my protein string, which is M, P, star, and S. And given, let's see, I wonder if I can actually run that in my REPL with the information that I have. Yep, actually it must be using a shorter string. Yes, it's using this shorter RNA string. So let me go set that. So given that, we have MP, star, and S. And so we can say, is the stop codon in AA? We've seen this before. It is a way of checking for list membership in this case. It's also a way to look for a string. So is it true that this is in MP star S? If I had that as a string, it's true. So again, the overlap between operations that you can do with strings and what you can do with lists are very there's a big overlap in Python here. So to do substring in a string or list membership in a list, you can use in. So now that I know that it is in, the stop codon is in AA, it is safe to therefore to say what is AA.index of the stop codon. It's 2. And what is AA at position 2? It is the stop codon. So what is AA up to position 2? It is M and P. And so and I want to stress that it's important to do this check first, because if I say what is the the index of X, that actually throws an exception. We want to avoid any operations that throw exceptions in our code, because I don't want to get into exception handling. So that is why it is important to check if the stop codon is in AA. If it's not, then there's no need for us to truncate it. So we can just we can skip this operation. But once we've done the check, we know it's safe to do the index. So that is why it is safe to then therefore overwrite AA with the string, the list slice of AA up to the index of the stop codon, and now we see that it's been truncated to AA, and we can turn that into a string by joining it on the empty string. So we get M and P. So that is what's going on with our translate function, and now our main function becomes significantly easier to read. We can, oops, let me jump to main. And we're going to translate the uppercase of that, and our translate function is also using our codons function that we've written. So they're starting to build up into more complicated programs. And let's see, are we passing our tests, both our unit tests that are built in here and our integration tests. And so if you'll notice when I run make test, one of the test programs is itself the prot.py. So it's looking in prot.py for things that look like unit tests, and it finds test translate and it finds test codons, and those are also passing. So that's the difference between just running PyTest manually and running make test. So now we have a really pretty easy to read program, and so just thinking about the next time I come to open this program and I want to read it, months from now, and I've totally forgotten how it works. One of the things I'm going to see here at the beginning, I'm like, oh, okay, this program takes a string of RNA. I know that. That's good. So I can kind of understand what get args tells me it's going to return args. And this is what's going to be at args. And it's very, very documented. I know what the fields are going to be and what their types are. And so when I look at main, which is always going to be the second function here for me, right after get args, I say, okay, I get my args, and then I'm going to translate the uppercase of version of the RNA. That makes a lot of sense. Okay, that's what this program does. Okay, what does translate do? Well, that's a lot of code. I'm having kind of a hard time understanding what it does, but visually I can see, oh, this is what's going to happen here. I see this calls codons. Okay, what does codons do? Well, codons takes a string, returns a list of strings. Still, how do I piece through this? Oh, there's a piece of documentation that tells me exactly how this function works. So this makes it very much easier for me to get. For the fourth solution, I want to get back into functional programming ideas with map, which we've seen before, but I'm going to introduce partial and takewhile. And to demonstrate this, I want to point out again how a list comprehension, which we've seen up to this point, how that can be rewritten as a map. So map takes as the first argument a lambda or some function that takes a single argument. And so, and in this point, we've talked about the star functions that I've used I think in the previous chapter to have a version of these functions that where the lambda can take more than one argument, but the plain vanilla map wants a single argument to be passed in to some sort of a function, and right here we're using lambda to create an anonymous function that's going to take a codon. So that is for codon. That's that part gets translated into the lambda definition. After the colon is the body of the lambda, and it really wants to be very, very short. It's something that fits on one line. If you start writing a lambda that has line breaks in it and continuations, PyLint and other tools are going to yell at you. And stylistically, it's very difficult to read a function that gets that long, where you're trying to cram in too much. So it would be better to write it as a defined, using a function definition. And you can use, as I've shown, you can use those defined functions instead of a lambda. You can use any function at all. And so this part of the list comprehension, which is essentially the actionable part of the for loop, the body of a for loop, that's what goes into the body of the lambda function. So I'm calling codon to AA get for that codon. And then the things that we're iterating over here in the for loop comes after the for codon in what? This is the thing here that's being iterated over, codons of RNA. And so that is the second argument to map, codons of RNA. So I can take that function that little function call here. Let me go back into my REPL that I've kept open this whole time. And this is another way of writing this idea. So we saw here, this was one way, and we look at AA, and this is in the other way, using a map function. AA, sorry. And they both give you the same answer. So why would you choose one over the other? I'm going to show you some things that you may or may not like, and you may choose to write code this way, or you may choose to write it the way that we've already written it, and that's totally up to you. So I want to introduce something that's similar to the filter function that we looked at, and it is a function called takewhile, and we're bringing it in from the itertools module, which we have used before. So if I go in here and from itertools use takewhile, it's going to take elements from a sequence while some predicate is met, which is exactly what we saw with filter. So it's just another way of expressing this idea. And if I execute that, I get again the same answer of what I saw before. And so why would I use that instead of filter? It's just another way of expressing the same idea. I'm going to take this a little farther and bring in another function called partial to show you something about called partial function application. And this is something that's very much out of functional programming. And I'm also going to bring in the operator module, which I brought in a previous chapter to look at the, what was the function? Well, I used one of the other operations in operator. I think I brought in add. And so I'm going to create a function that is called not stop. And it is going to be a partial application of the operator not equal. So let's look at what is the help for operator not equal. It takes an A and a B, and it will tell us if A is not equal to B. It's the same as writing A is not equal to B. So if I put this in, if I say, let's just say operator not equal 4 and 4. That is false. Those things are equal. Operator not equal 4 and 5, that is true. Let's put in two literal strings, A and B. That is true. Those are not the same. A and A, that is false. Those are the same. So that's how operator not equal works. If I want to say, what I want to say is something is not equal to star. So I want to essentially bind that first argument, star, the literal string star, to this operation. And I can do that with what's called partial function application. So I can create a function called not stop that wants it already has the star bound to it. So if I pass star, it's going to say, is star not equal to star? Well, that's not true. That's false. It is true that A is not equal to is not a stop character. So I have now created a function that takes one argument and applies this operation of saying operator not equal to the argument that it already has bound. So that means that I can rewrite my list comprehension, my takewhile, with this function. And now it just starts to read like English. If I understand what's going on here, I'm saying, oh, take from, while it's not a stop character, take all the elements from AA. That's kind of cool. It reads really nicely. So how can I use this in my function? So I can change if we go back into my protein function, and I change this line here where I have written it as a list comprehension, and then I do this indexing. If I get rid of those lines and instead I use my lambda, first off, I'm sorry, I use my map to another way of writing this, and if you'll recall, I'm not putting list around this, so this is actually just going to create a generator that is a lazy function that hasn't yet been turned into a list of amino acids. And now I can now use on my generator, I can take from that generator, I can take while it's true that the elements that it returns are not equal to star. And so what's going on here is I've essentially, when I said earlier that, with the list comprehension, that we do turn the entire string, the entire RNA string into a list of amino acids, and then we truncate it. This is actually using lazy functions so that AA here is now a generator that has not generated all of all the codons, all the amino acids. And so this take while I was kind of leading up to, why would I use take while over filter? Well, because this is a way of lazily evaluating this generator to only take while the values are not the stop, and then to stop taking from the generator, and that will return a list that then gets turned into a string. So a lot of kind of heavy lifting going on behind the scenes to really to be much more computationally efficient and only produce the values that we need. And so if I go run my make test, this should not change anything. Oops, we actually have a way. So this is why we run our tests. I need to, let me go look at the solution 4, and I need to import a couple of, I need to import a couple more, I need to import those functions. So that is why we have tests. Operator. Import operator. That is why we have tests. And now we're passing all of our tests. So this is, why lean into functional programming? Well, it can actually be more computationally efficient because of the idea of lazy functions that don't produce all of the values right away. It also, to my understanding, to the way that I've come to write code, this looks very readable to me. Reads like English. So I would probably, if I were going to write something like this, I would probably write it like that. However, there is in the Bio.Seq class that we've used many times now, there is actually let's go back and put our RNA string to this. There is a function that will do everything that we want to do. It's called translate, and that should not be a surprise. So when you are actually writing production level code, you're going to be faced with, first off, you're going to have a time. There's always going to be, you have a limited amount of time to get things done. And so you're probably going to Google for a solution that already exists, and that is, generally speaking, the way to go. Maybe you find something on Stack Overflow, or maybe you're using some sort of coding agent. If you have the time to invest in writing your own function, you will probably learn something. If there is an off-the-shelf tool, though, that will do what you need, you're probably going to use it. Although I would hope that you would have the time to at least verify that you understand the code and that you have some assurance that it's actually correct. In this case, it's such a straightforward thing to do, and so many smart people have given of their time and energy to write the Biopython module. It really makes sense to just use Biopython for this. And so this is the version that I would recommend that you use. However, there's something that we need to take care of. The output should not include the stop codon, and I mentioned this earlier. So if we, for instance, translate this one where the stop codon is in the middle, we don't want to see anything past that. So there's actually an argument to this, as you would imagine. You only want to go up to the stop codon, and it's called toStop, and it takes a Boolean value of true. And if you pass that, then we can actually write a version of our program that gets rid of so much code. We can actually do this, Therefore, we don't need takewhile, we don't need that. We need from bio import seq. And then essentially our code just got, let me get rid of all of this, all of that. Now our program is so very, very short. And if we, let's try our tests, make test. There's still a problem. It doesn't, because we don't need lists now. Pylint is saying we don't need that import, and that's fine. I'm happy to remove it. And now our program is so very, very simple. And this is obviously the solution that I would recommend that you actually use if you were writing production code. And so we come back always to the idea, we've written so many different solutions here. Which one is, well, at least the fastest? And I have in this directory here a bench program. It's actually set to 1,000 iterations in the GitHub repository, but I lowered it to 100 because it was really takes quite a while. And so I wanted to run this program to see which one is the fastest. And it comes up with the list slice is only really slightly faster than any of the other versions. The Bio.Seq actually comes in as, well, quite a bit slower, maybe not statistically enough that I would still probably choose that over the other solutions just because it's such standard code, I guess, unless it was really proving to be some sort of bottleneck in my computation. But it is nice to know list comprehensions are pretty fast, faster than doing map takewhile by only very, very, very little bit. So it's good to know, it's good to have this information. And so I will leave the bench program here for you that you can run yourself and prove on your hardware which seems to be the fastest when I publish the book as the same as it is now. The fastest one is close to 1.5 times faster than the Biopython version. So I mentioned it a little earlier. I mentioned at the beginning that we are only handling one of the frame shifts, so we're starting at 0. But you could add a frame shift argument to your program that allows a person to choose something between 0 and 2 inclusive, and or you could do 1 and 3 if it makes more sense to you as a biologist, and then correct for offsets. And therefore, you could, instead of reading from 0, you could read from a different frame shift, from a different frame, and produce other protein strings. And that would be the first thing you would want to do is add data to create the expected outputs for the other 2 frames, given your inputs. And so you would be a fantastic exercise for you to build that for yourself. This chapter really focused on a lot more on writing and testing and composing functions. So we ended up now with 2 functions that we're able to write and test, write unit tests for, and then to compose them together. And that is how your programs are really going to go as they get longer and longer and more complex. And so that's why I'm teaching you these skills to write small programs and tests. Some of the other things that we talked about, k-mers, that's actually going to be important in the next chapter. So we're going to be using k-mers to look at subsequences. And so a k-mer is a k-length subsequence of some other sequence. And k-mers, by definition, do overlap. But codons are 3-mers that do not overlap, and they come from a particular reading frame, although there is something called dual coding exons that actually skip into a different frame in the middle of translation, if you can imagine that. We talked about how dictionaries are useful lookup tables, and we did that, and we used those for translating a codon into the amino acid. We again leaned into how for loop, a list comprehension, and a map are all basically the same idea. We talked about how takewhile is similar to filter, And the reason for using takewhile is that it worked with our lazy map evaluation in order to not actually produce all of the translated codons. So and in that takewhile, we also used a partial function that did a comparison against the star to know when to stop reading at a stop codon. And finally, we just said, well, okay, there's actually a Bio.Seq.translate function that does all of the work for us, and that's good to know, but now we know what's going on underneath the hood.